DNA structure. DNA is a long chain in which is located on every cell of our body. It is formed of nucleotides. The DNA information is stored from four nitrogenous bases: adenine, guanine, cytosine, and thymine. In every nucleotide contains a phosphate, a sugar, and nitrogen base. In every sugar, there is 5 carbons named deoxyribose. And actually, the order in which the nucleotides are organized is important, just for the reason that the order depends on genetic code. Cytosine and guanine and adenine and thymine are bonded with hydrogen bonds. Adenine and thymine have double bonds, and cytosine and guanine are triple bonds. The structure of the DNA is a double helix, such as a twisted ladder. This set of DNA structure have the same amount of nucleotides that the RNA. The difference between the RNA and DNA is that DNA is a double helix and RNA is a single helix. The DNA contains the instructions that our organism needs to continue working as normally it does.
DNA replication. DNA replication is a very important process, and it happens every 8 hours in your body. It all starts in the cell cycle. To be precise, in the interphase, not in the mitosis. It happens inside the S phase of the interphase. The first thing we need to know is that we will split the DNA with the help of the helicase. Also, the proteins will take care of not letting the DNA get together again. The two strands will be leading and lagging. Then, we will have to name the primes. We have from 5 to 3 and from 3 to 5. This consists on the position of the sugars. Counting clockwise, we have the 1, 2, 3, 4, and 5 outside. And if it's upside down, 1, 2, 3, 4, and 5. Now, the primase produces a primer to start synthesizing the DNA. Now we have the polymers, which walks by adding the missing and exact bases to the DNA strand. But here it gets a little tricky, because the polymers can only walk in one direction and entirely complete only one strand. We have the lagging, which goes from 3 to 5. This means that the polymers, as long as it walks on by, it will leave some chunks along its way, which will be called Okazaki fragments. After all this process, it comes the ligase, which finally bonds all the bases with its due pair. Adenine with thymine, which are double bonds, and cytosine with guanine, which form triple bonds. And that's it. Now we have 2 new brand strands of DNA.
Transcription. Transcription occurs inside the nucleus. And during this process, the double helix is split into 2 separate strings by DNA polymerase. In the DNA contains genes. Genes are the piece of string that contains nucleotide. While RNA is formed, it is just required a single strand of DNA as a template. As the RNA strand detaches from the DNA gets back together. The piece of mRNA moves into a ribosome. To start the translation process, there are three types of RNA. mRNA is a piece of information that moves into a ribosome and later from a protein. rRNA is part of the ribosome where the protein will form.
And tRNA comes from the cytoplasm and attracts the amino acids into the ribosome to develop proteins. Translation. Translation is the process in which mRNA is converted into a polypeptide. Let's remember, one or more polypeptides make up a protein. First, the RNA polymerase will always know where to start. They will always be a starting codon and the stop codon. Then, the small ribosomal unit will join to the mRNA strand. The large ribosomal unit, which has three sections, which are E, P, and A, and it attaches to the small ribosomal unit. Then, the base pairing occurs between the mRNA codons and the tRNA anticodons. A codon is a 3-nucleotide sequence that codes for an amino acid. Codons are read without spaces as a series of 3, and it is called the reading frame. The elongation process is in which various amino acids are brought through the mRNA strand, and it will stop until the stop codon is reached. As the large subunit advances, each section changes. As the eTRNA advances, a new amino acids enter the A section, and one advances to the P. This process will stop until it reaches the stop codon. And that's it. Now we have a new polypeptide formed.
Gene expression and regulation. Lack operon. All of this process occurs inside an operon, which is made up of a promoter, a segment in the DNA which will define where the RNA polymerase to begin the transcription process. An operator. This is another segment in the DNA which later the repressor will bind to, blocking the RNA polymerase from moving forward. And gene sequence.
In this process, there will always be a regulator gene, which will regulate and control the activity of one or more genes. For example, the repressor is made by the regulator gene, so they can save energy.
The promoter will bind with the RNA polymerase and the operator with the repressor. During this attachment, the RNA polymerase won't be able to transcribe it because the repressor blocked. Until a sugar, known as lactose, binds with the repressor, distracting it, and make it moving from the operator, allowing the RNA polymerase to pass to start the transcription process.
Introns and exons. In eukaryotes, genes are composed of two elements, which are introns and exons. First, let's learn what is an intron and an exon. An exon is a region of nucleotides which will code for amino acids in proteins. And an intron is a region which will be discarded before translation. Introns are not necessary for the process to make a protein. That's why they are removed from the mRNA strand. First, we will have a strand of exons and introns. Then, the introns will be discarded of this chain. All the introns can be recycled into new nucleotides to make another mRNA strand. And after the introns are gone, the exons will join to start making a protein. In eukaryotic gene, we also have enhasers, which will influence the transcription process. It also includes transcription factors, which are regulatory proteins which will interact with promoters, enhasers, and RNA polymers. To regulate transcription in eukaryotic genes, activators usually bind to the enhasers, and other transcription factors bind to the promoter. These transcription factors helps the RNA polymerase get in the right position to start working. Normally, the activators are far away from the gene. That's why a loop is created in the DNA. This loop will make the activator and the transcription factor interact with each other, increasing the transcription of that gene.